Bueno, voy a presentar a Brandon, pero primero me voy a presentar yo porque no sé si aquí me conoce todo el mundo. Yo soy Conchi Muñoz Díez, trabajo en el departamento de agronomía y hice la tesis doctoral allí en variabilidad genética en olivo y durante los últimos 4 años he estado trabajando en la Universidad de California en Irvine, en el grupo del profesor Gaut. Entonces llevamos 4 años de colaboración y para mí es un placer que él aceptara la invitación de venir aquí. Ha estado 2 semanas para conocer la Universidad de Córdoba y un poco para seguir la de colaboración porque estamos intentando iniciar algo de genómica en olivo. Shall I translate for you?
No, that's okay.
Bueno, ahora esa soy yo. Ahora Brandon, creo que todo, bueno, se mandó la información y tenéis acceso a su currículum. Brandon es profesor de la Universidad de California en Irvine desde 1998 y es uno de los referentes en genómica de plantas, en evolución, en proceso de domesticación de plantas. Y últimamente, bueno, en los últimos 5 años iniciaron una carrera muy interesante en evolución experimental de microorganismos. Concretamente llevan un experimento de larga duración donde están viendo evolución a tiempo real en E. coli, en Escherichia coli. Y bueno, y además de ser un gran científico es mejor persona y durante estos 4 años yo creo que no podía haber elegido un sitio mejor donde ir y nadie mejor con el que colaborar. Así que para mí estoy súper contenta de que él esté aquí y de que pueda dar esta charla. And I guess now is your turn.
Okay.
You have to talk.
I hope you said something nice. So, thank you for coming. I want you, before I begin to talk about science, I want you to know 3 things. The first is I have to apologize that I don't speak Spanish. You know, in California, just as many people speak Spanish as English. And in fact, I'm trying to learn a little Spanish, pero soy viejo. Y mi cerebro funciona lentamente. So I'm trying to learn. I also want you to know that I'm actually incredibly grateful to Spain, the country. And I have 3 reasons for that. And I'd like you to know about them. The first reason is Granada, because this is my 25-year-old son, and he spent a year studying math in Granada. The city was remarkably welcoming to him, and it was a fantastic opportunity for him, and I'm told he speaks Spanish with an Andalusian accent. And we actually visited him in the summer before he left to return to Irvine, and it just so happened it was during the World Cup, and we were lucky enough to be in Granada during the World Cup final. So that was actually very exciting for me. And it's important that you know, as a United States citizen, I will probably never see our team win a World Cup. So it's nice to see Spain. The second reason is Bilbao. That's because my youngest son, Jared, is living in Bilbao, playing water polo and teaching English. However, I will say the Oficina de Extranjeros has not been very nice to him, so he had to go back to the United States last week to try to get a long-term visa. And it's kind of fun. It was fun to see him. I saw him 3 weeks ago in Bilbao. It's a very different city than Cordoba. I will say that the players on his water polo team find it very hard to say his name, Jared, so they call him Tor. And then the third reason I'm very grateful to Spain is Cordoba. So I've been here almost 2 weeks. Everywhere I go, people have been very warm and welcoming. But that's not really why I'm grateful to Cordoba. I'm grateful to Cordoba because Conchi came from Cordoba. And it has been an absolute privilege, and I think she knows that, to have her in my lab. And I will say, yesterday was Thanksgiving. I've never been away from my family for Thanksgiving. We were going to be in Bilbao. It's a long story, but I now have a Thanksgiving family in Cordoba because these kind people put on a Thanksgiving dinner. So, fantastic. I'm grateful to Spain. I'm also, the last thing I want you to know before I begin, I'm an evolutionary biologist. Everything I do is through the lens of evolutionary biology. And I'm going to talk about some very different kinds of experiments today, but the common theme is evolution. So the title of my talk is Evolution as an Experiment, And I'm going to talk about one huge experiment, one not so huge. And there's a common theme to them that I'll explain. So the first experiment I want to talk about is the domestication of crops. That's not an experiment that you or I have done, but it's an experiment that humans have done through the ages, and of course all over the world, from naranjas in China to fresas, did I say that properly? In South America. Lots of domestication has occurred. So I'll talk first about domestication, and then I'm going to turn to an experiment that was in my lab, an experiment having to do with E. coli. So what's the common theme between these experiments? Here's the question, the main question I want to ask today. How replicable is evolution? Is evolution very flexible? It can find many solutions to one problem? Or is it sort of channeled so that to adapt to a new condition, only a few sets of genes really work? Hopefully this idea will become more obvious in a minute. So let's start with domestication and talk about what domestication does. So I think probably many of you know that Brassica oleracea has been domesticated into several different kinds of crops, from cabbage and Brussels sprouts, kale, broccoli, cauliflower. And they have one thing in common, they're all disgusting. I don't like any of them. But we've had this domestication event that has made huge morphological, phenotypic changes through domestication. Sometimes domestication doesn't have as big a phenotypic effect as in the case of the Northern American sunflower. Nonetheless, there have been important changes. There's fewer flowers in the domesticate, there's bigger seeds, and more oil content. So even these cases where there aren't big changes, aren't big, obvious changes. There have been important changes. So some time ago, someone came up with the term the domestication syndrome. And this is true of all domesticated plants, that these kinds of events occur. Larger fruits and grains, often fewer fruits. The plants are more robust, there's less branching, and so on and so on. And this is true of any domesticated plant, from oranges to strawberries to sunflower. Now probably the most interesting, and I'm biased, the most interesting domesticated plant is maize. And it's interesting because of the huge morphological change that has occurred. So until the 1980s, we weren't even sure where maize came from because it is so different from its original plant. The original plant grows in Mexico. It's called teosinte. And this is not a very good picture of teosinte, but when it's by itself, it forms lots of branches. And these are the seeds. These seeds are only a single row, and you can see they're sort of triangular seeds, and they're very hard. They're so hard you can't cut them with a knife. They are basically inedible, unless you treat them. But now we have corn, that all of you know. We all know this corn ear. Ears of corn do not exist in the wild. That is something that occurred during domestication. In addition, corn does not have lateral branches. It doesn't branch. So all kinds of changes have occurred in corn. And of course, they're still changing. Breeders are trying to improve the yield of corn until someday I think it may look like this. Now, I got interested in corn when I was a PhD student. And I became interested in corn from the viewpoint of evolution and population genetics. And when I was a PhD student so long ago, 1992, this was one of my very first papers. And what was exciting in 1992 is that PCR was a new technique. And for the first time in history, evolutionary biologists could sequence genes and then sequence it from another individual and another individual and see how individuals varied on the DNA sequence level. This was very, very exciting for us, our first vision. In fact, this was the first paper to be able to do this in a gene in a plant. So it's kind of exciting. And as evolutionary biologists, oh, by the way, there were 8 sequences of one gene in this paper. That's all there was, but that was a lot at the time. So we could do things like estimate this parameter theta. Theta is just a measure of diversity. How much diversity is in the sequences? And the nice thing about this measure theta, there's lots of nice things about it, but it just summarizes how much variability there is within a system. And if you look back on this paper, it's sort of unremarkable, except for 2 things. The first is, we could see these different sequences didn't cluster by species. That is, if you built a phylogeny of these sequences, they didn't cluster by species. That's not so important. More important is the fact that nucleotide variation, the amount of DNA sequence variation in corn, a domesticated plant, is very, very high. In fact, much higher than sequence variation in humans, by about an order of magnitude. Why is that strange? Well, let's think about what happens during domestication. You have a wild plant like teosinte, wild corn. It has lots of genetic variation. But when a plant is domesticated, not the entire population is domesticated. Just a few of the plants are chosen for domestication. So that means there's a bottleneck, a narrowing of the genetic diversity, so that the domesticated plant should have very little diversity. Okay? And yet here there's corn with a great deal of diversity, much more than humans. So the fact that it has so much diversity suggests that the size of this bottleneck during domestication must have been very large for genetic diversity to come through. So when I finally got my own lab, that was one of the very first things I did, is try to figure out how big was that bottleneck during domestication. How many individuals were domesticated? And to do that, we used some fancy modeling techniques called coalescent theory. And what was interesting about this paper is no one had ever tried to do this before. That is, try to understand the past history of a bottleneck in any kind of population, including humans, using DNA sequence data. So actually, plant domestication was the first place that did this. What we did is we ran these models, and it's not so important the details, but we basically tried to understand if there was an ancestral population and the domesticated population, how big was this bottleneck? So we could estimate these functions and estimate the size of a bottleneck. So for example, we know from archaeological data that the longest that bottleneck could have been, domestication, was around 2,000 years. And if that was true, then maize domestication was based on a few thousand individuals. So we were just trying to get a sense of how many individuals could contribute. Okay, so basically what I was trying to do is find how big is this? How many individuals could be domesticated? But it became apparent that something else very exciting is going on. That is that there's different kinds of genes. There are genes that don't contribute to important traits, and then maybe there are genes that contribute to the formation of the ear, for example. And those genes are very precious genes. Those genes would be selected. There might be one allele that makes this important contribution to the ear. And so, while some genes will have a domestication bottleneck, other genes will be highly selected so that there's only one variant in the crop. And this gives you the idea that you can actually try to find these genes. Find these selected genes using population genetics. Now, why is that important? Throughout plant breeding, we've always used what's called a top-down approach, where you start with an interesting phenotype, the corn ear. You do QTL analysis or association matching based on that phenotype. You find interesting genes, and then maybe you do some population genetics. So you're trying to find the interesting genes. Well, this suggested that we could actually do a different kind of approach to find the interesting, important genes. That you start by trying to find selected genes using population genetics, and that tells you which genes might be important in the morphological change. So, for example, from teosinte to corn. So, we did that. This is a long time ago now. All of this stuff was very long ago. We, in fact, tried to find all the selected genes in maize. Now, when we did this study, we couldn't sequence whole genomes. This was before the whole genome era. And so what we tried to do is estimate how many genes are neutral and how many genes are selected. These are the important genes, potentially, for transformations during domestication. So we did all these details, estimate demography, blah, 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 blah. But the important point is we found a number of genes, and we estimated that 2 to 4% of the genes in the entire genome had been selected during domestication. And so if we extrapolate, we only had 800 genes in our study, but that was a very large study at the time. We estimated there were 1,200 selected genes. Okay, cool. So what are these genes? We could make a list of the genes. But you know what? A list isn't very satisfying. So we had all these genes, and to be honest, once we finished this big project, it took about 10 years to develop the methods and the statistics, to generate all the data. I ended up being not very impressed. I don't know if you know this picture. This is an American gymnast who just got the silver medal in the Olympics. And she got the silver medal even though she was by far the best in her event, but she made a mistake. And so she wasn't very impressed with her silver medal. So I wasn't very impressed with my list of genes. And you can do these clever things. So for example, we know where there are QTLs in the genome, and you can ask, do our genes that we think are selected, that would be on this axis, do they correspond with QTLs? And we found they did. Well, that's really neat. But still, I was sort of unimpressed. We could also show that the genes that we think are selected were actually upregulated in the ear. So if they're important genes for the transformation of domestication, maybe this is an interesting set of genes. But still, I was a little bit unimpressed. And here's why I was unimpressed. You can get a list of genes from population genetics, but you can't stop there. So what do? One thing you can do is if you have to verify that the genes really are interesting, and then you have to go to some very difficult genetics, where each gene might take several years to decipher. So I decided not to do that because I'm impatient. You could also revise the models and methods, and I haven't given you the details of those models and methods, but the honest truth is, I'm not a great theorist, so I didn't think that would be the thing to do. So the other thing that I think is really interesting is to look at more genes and more crops. And now we're coming to my theme. That theme is, how replicable is evolution? If you use the same approaches in other crops, do you find the same sets of genes? That is, does evolution give you the same sets of solutions? Right? So in just a minute, I'm going to compare between species to see if we can answer this question. Does evolution come up with novel solutions, or does it use the same old solution over and over again? But I must say, this was several years ago, and I didn't get funding. And so I had to wait a little while until there's enough data to answer this question. So I want to take a brief intermission just to tell you that I have been doing things. It'll be very brief. So one of the things that we've been very interested in is things like the epigenetic effects on genome evolution. And we've been very interested in how methylation of DNA affects gene expression. We've also been interested in other kinds of variation, not just at the DNA sequence level. So this is a map of Mexico, I think you know. And we did a sampling trip. And when I say we, Conchi did more sampling than I did. And we went to get both maize and teosinte, its wild relatives. And that's proof that Conchi actually did go sampling. In fact, she told me the other day you spent a night in the city where they had that, I won't say it. So, one of the very interesting things about plant biology is we know so very little about so many things. And one of the things we don't really know about is how genome size varies. So we went to Mexico and found all these different populations where we measured genome size. And when we got back to the lab, Conchi grew the plants and she measured the genome size. And what I want you to see from here is that we have both cultivated corn and wild teosinte on this. And you can also see the genome size is scaled so that between these two extremes, there's about a 30% difference in the genome size in the exact same species. So then the question becomes, oh, by the way, something very interesting about this result is that if you just take the environmental information, temperature, mean temperature, rainfall, all those kinds of things, and build a linear model, you can actually predict genome size very, very well. So genome size is something that varies a great deal, and it's related in some way to environmental conditions. Beyond that, we really don't know. But one thing we are very interested in is what causes this genome size variation? Why is one genome 30% larger than the other genome? What is it in those genomes that are changing the size? So what we did is we took a control line and two very extreme lines that differed in genome size. And this is all Conchi's work. And they differed in size from basically on the normalized scale to the reference, 26% higher and 2% lower. And Conchi did a bunch of work with small RNAs and also was sequencing the entire genomes to try to find out what is this difference in genome size. And I don't have the time, I want to get back to the theme of my talk, but I do want to say that we found some really interesting things. So for example, genome size variation is not caused by transposable elements. And that's what I assumed would be the case, because transposable elements can proliferate very quickly. But in fact, it has something to do with heterochromatin. We don't know how this is happening. We know that transposable elements fall into three discrete groups that are shown here that have very different evolutionary dynamics. It's a mystery. Anyway, I will say, while I've been waiting to study crop domestication more, I've also had the fun to do some work in olives. And I have to say, it's been fun because it's really been work that's based here in Cordoba. Conchi was nice enough to let me contribute to the paper, and it really was the most fun paper I've ever worked on. And I only mention it because it's online this week, so if you want to see Conchi's new work, there it is. Okay, so I waited for years and years to wait until we have more genes sequenced from more crops. Are the same genes selected in different crops? Now there's been publications that we can make this comparison. There's been selection screens of the very type that we developed in my lab on maize, soybean, rice, common bean, cucumbers, and more. So for example, my former postdoc, Jeff Rossabara, redid our work in maize, only now with whole genomes. Funny enough, he found the exact same thing we found. There's about 1,200 genes under selection, or 2 to 4% of the genes. Now in rice, there's also been really a very nice study published in Nature in 2012 I found 55 regions under selection. 1,200 versus 55. The question is, do these two correspond to one another? Is the domestication solution in rice the same as the domestication solution in maize? And you might say, that's not a fair question. Rice and maize are very different. Well, there's many, many people that argue that the grasses, both of these are grasses, is an interesting system to make these comparisons. So I think most people would say it's fair. So what happens when you compare rice and maize? Are the same genes under selection? Well, I did a little analysis. I found 1,526 genes were in maize. How many selected genes in rice were also selected in maize? The answer is 4%. Okay. In case you're wondering, that's the random expectation. Okay? What that means is, we've seen selection in maize, we've seen selection in rice, and they seem completely unrelated to each other. That suggests a couple of things. It might suggest that evolution really is very flexible, with very little constraint. That there can be all kinds of solution to a similar problem, domestication. But we have to remember there's all kinds of complications with these data too. In fact, some of these studies I don't trust, I'll be honest with you. Okay, you say, I don't believe rice and maize. That's too far of a comparison. Well, how about I do this? This was published this year in 2014. Common bean. There were 2 domestication events in common bean. The same species. One domestication was in Mexico. One domestication was in South America. Were the same genes selected during domestication? So, this group did screens on both domestication events. They said they found 1,835 genes and 750 genes. There was only 59 genes in common between the 2 events. That is, again, the random expectation. So again, we have to ask, is evolution really that variable? That even within the same species, you can domesticate a species twice, and different genes are involved in the changes in domestication? It's not clear. Let's take another approach. Remember I talked about top-down, where you start with a phenotype, and this is a very, very interesting gene that relates to shattering in sorghum. So if you don't have this particular mutant, it shatters in the wild, but then you get the mutant and sorghum seeds, or the grain remains on the stalk. It's very important for domestication. So this gene is a Mendelian trait. It was isolated via fine mapping as a QTL in rice and maize. So maybe this gene does play an important role in sorghum and rice and maize. And in fact, it seems to be a gene that was selected in rice. Maybe evolution does use the same gene over and over again, and maybe the other approach just isn't very good. But here's the interesting thing. We know from QTL studies that it's not a major gene, shattering one, in rice. There are other genes that cause the lack of shattering in rice. And we also know that it's not implicated at all in wheat, the lack of shattering of wheat. So it's very interesting. We have this gene. It has been selected in parallel, but it doesn't play a big role in all the plants. It plays its biggest role in sorghum, a smaller role in rice, and no role in wheat. So it seems like evolution is not that replicable from what we've seen here. So I'm not 100% convinced by everything I've shown you, because there's all kinds of underlying assumptions to the work that I've compared. So what you do when you can't answer a question that you want to know the answer to, and this is the question, how replicable is evolution? You go to a new system. And so I, with some collaborators, have gone to bacteria. We're asking the exact same question. How replicable is evolution? And we're using an approach called experimental evolution. And it really was made famous by a guy named Rich Lenski, who's been evolving E. coli in his lab since 1988. That means someone goes in his lab every day and messes with the E. coli since 1988. No Thanksgiving, no Christmas, no weekends. Somebody has to go. And the design of his experiment was very, very simple. Essentially, he took this thing called E. coli B, and he put it in a media that had limited glucose. So he was selecting for bacteria that don't need a lot of nutrients, that are very efficient. He put it in 12 different lines. I only show 4 here. So he had 12 replicates of his solution. And every day he lets them grow, and then he dilutes them into new media. The next day, the next day, the next day. And they've done that every day again since 1988. Okay? Now, why would somebody start an experiment like that? Well, one of the great things about E. coli is that you can easily judge whether they've adapted or not by measuring something called relative fitness. What this means is you take your evolved bacteria that you've been evolving every day, and you take your old bacteria that you started with, that's in the freezer, you let them grow together, and then you see who wins. And you can easily count who wins by counting colors on a plate. This one is one color, that's another color, and you see which one grows better, which one outcompetes the other. And that's called relative fitness. It's a fitness relative to where you start. And in Rich Ledsky's case, you can see that fitness, the ability to grow in low glucose, increased very rapidly. And he was particularly interested in the question of replicability as well. You have 12 replicates. Do they all increase in fitness in the same way? I'm going to skip that. So we know very well his experiment. It's very famous, and it was started at UC Irvine, as a matter of fact. And we thought, that's great, but we need to do this on a bigger scale. And we need to include DNA sequencing. So I and 2 of my collaborators at UC Irvine did the exact same experiment. We started with actually a Richard Ledsky line. We put it in the same media, and we let him grow. But there's some important differences between the experiments. One is we put it at high temperature. So that's the selection. We're selecting for high temperature, bacteria that can grow at high temperature. And we did it on a much bigger scale. Instead of 12 replicates, we did 115. And we did this every day for a year. Now you may ask yourself, who has to do that work? Who comes in every weekend and every holiday to play with that bacteria? That's my wife. She does it. She works in my lab. She didn't like me for 1 entire year when this experiment was going. So at the end of the year, we had 115 evolved bacteria. They've been evolving for a year to high temperature. Their relative fitness on average was 40% better than when they started a year before. So they clearly had evolved to high temperature. But what we really wanted to know is how replicable is evolution? You have 112 or 115 of these experiments. Do they all evolve to high temperature using the same genes in the same way? So to answer that question, we had to sequence all of the genomes. So we sequenced 114 clones from 114 different replicates. We found all these mutations, and here's a collaborator who was very key on this paper. This is a picture of each of the genomes. So there should be 114 genomes here. And these colors represent different kinds of mutational events that occurred during evolution. So if you look at that, there's always one thing that I see very clearly. That is that many, many, many of the different 114 bacteria contain this mutational event. That, it turns out, is the deletion of some genes. And clearly it happened very often. But as you step back again, it's a little harder to see many more patterns in what occurred during evolution. Maybe you can say, I see something here, maybe something here, but there's also a lot of random changes. And I want to say, we think all of these changes contributed to adaptation because of the conditions of the experiment, which is large population size and all these other things. So let's look at this data in a little more detail, again trying to answer the question about replicability. So how replicable is evolution in E. coli? It depends a little bit on the scale that you look at it. So we might ask the question, do you find the exact same mutation in different lines? And how often out of 114 lines do you find that mutation? Or you might say, do you see a mutation in the same gene? And how many times out of those 114 lines does the mutation occur in the same gene? Or you might even say, maybe there's a protein complex where proteins interact. How often do you see a mutation in the same protein complex? So let me show you that data. Here is how often you share a point mutation, the exact same mutation, 2% of the time. Very rarely do you see the same mutation in different replicated lines. But that's not to say that some mutations aren't found sort of commonly. So for example, there's a gene, RNA polymerase. 18 of the 114 lines had a point mutation, a change in base pair, with the same codon. Okay? There's something called the Rho terminator. 17 lines had a point mutation in the same codon. So you can occasionally see mutations that have occurred in more than one of these replicates. What if you look at the level of genes? Well, you can see about 20% of the mutations in genes, if you look at the gene level, are shared. So many more. And actually, sometimes, some genes are mutated a lot. So this gene was mutated in 65, or more than half of our lines. What's interesting about this gene, it was almost always an insertion or deletion event that knocked out the gene. Okay? So it wasn't the same mutation every time, but it was the same gene. One of the nice things about having a highly replicated experiment is you can do some very simple things with the data that can be very interesting. And this actually, I find absolutely amazing. So what you can do is simply choose data over and over again. Bootstrap resampling. And what you can do is choose a different number of sequence lines. Okay? So for example, I might choose 20 out of our 114, and then estimate some things about it, like the number of different mutations. And what I want to show you here is, if you, say, take 20 individuals and ask how many genes are mutated, it's about 50 genes. If you take 40 individuals, 60, 80, 100, you can see it plateaus. In our data set, there's never more than about 60 genes that are mutated. Okay? So that means that really evolution is somewhat constrained. There's 4,000 genes in E. coli, and we really only ever see mutations in 60 of them. Okay? But here's the thing I find completely fascinating. If you say, how many mutations do you find? Our curve never plateaus. We would have to have a much bigger experiment. What does that mean? What that means is we're having, at this point, an unestimable number of mutations in a small number of genes. Wait, what does that mean? That means I naively thought if you have an important enzyme, you might have 1 or 2 sites that can change and maybe lead to an adaptation. What this experiment implies is that evolution is far more flexible than that. It may only have a set number of genes that can mutate, but within those genes, there's a large number of sites that can change and change the protein so that it adapts. I hope that makes sense. I think it's really neat. How much time do I have? Yeah, quite a bit. Okay, now there's something else that's very interesting about this data. Remember, we have 114 DNA sequences of the whole genome. And what you can do is you can make a graph like this. We call it the co-occurrence graph. It looks very complex, but it's actually very, very simple. The idea here is you have a gene, CLS, and then you have another gene, ICLR. Actually, these are sets of genes. If there's a dot in that square, it means that at least one genome had a mutation in CLS, and ICLR. Okay? All right, hopefully we get that, and now you can look at the square. And there's some things that are very obvious. The things that really jump out to me are empty squares. That means you never ever see what this means. This is in one gene. You never see two mutations in that gene. You never see a co-occurrence of mutations. Now, this makes perfect sense for this gene. I told you about this gene. It is YBAL, and in over half of our samples, there was a mutation in the gene that inactivated it. So why would you only get one mutation in this gene and not two? The reason is, once you've been inactivated, it's inactivated. If you mutate it again, it's not going to be an adaptive mutation because it's not doing anything, right? So this makes perfect sense. There's no mutations. That is, you don't get two mutations in that gene. We never saw them. But then there are other strange things that we really cannot explain. This here is rod. Rod refers to the shape of the cell. There are 6 known genes that affect the shape of the cell. These rod genes were mutated quite often, 54 times in our experiment. But never did we see more than one mutation in these genes. And that's statistically improbable if you do the right statistics. So for some reason, once you get one mutation in this set of genes, other mutations don't show up. That's what we call epistasis. There's a negative interaction among these things. But we have no idea why that's the case. It was completely unexpected. And then we have this gene along the diagonal. You'll see it's the only gene along the diagonal that has circles in it. That's because this is RNA polymerase. RNA polymerase was the most mutated gene in our study. It was the only gene that had more than 2 mutations in any clone. And it had 86 total mutations. And we don't know why you might get 2 mutations in this gene, but one idea is you have one mutation, it changes the RNA polymerase. Some of that change is good, some of it's bad, and then you get another mutation that compensates for the bad part. We're actually studying this. I'll tell you a little bit about it in a minute. What this co-occurrence graph shows us, though, is that there's some very interesting things going on. What we can do is measure something called linkage disequilibrium, and that tells us about the association of mutations. And what this graph is showing us is where the graph is dark in red, is showing us where you tend to have positive associations. So what this means is, if you get a mutation in RNA polymerase, you also tend to get mutations in these rod genes. In fact, you get more mutations in both genes than you expect by chance. There's some sort of interaction we do not understand. Also, so for example, this mutator gene, terminator gene called Rho, if you get mutations in it, you also tend to get mutations in another gene called CLS, and we don't know why. But interestingly, when you look at the whole graph, it appears there's 2 ways to evolve to high temperature in our experiment. One way is to have mutations in RNA polymerase and all of its associated genes. The other way is to have mutations in the terminator of RNA polymerase and its associated genes. So what we found is that there's 2 pathways. 2 pathways to evolution in our experiment. We don't know why. It's very interesting. It's very neat. So what do you do after that? You say, okay, there's 2 pathways. One is, if you get evolution in RNA polymerase, you tend to have these other genes involved. The other pathway is if you get this Rho terminator and you get mutations there, you have these other genes involved. What you might ask yourself is, one pathway better than the other? If you're an E. coli, and I know you're not, but if you're an E. coli, would you rather, is it better to evolve by the left-hand pathway or the right-hand pathway? Well, I can tell you this, you can measure fitness, and at 42 degrees, we can't see any difference between the E. coli clones that evolved this way and the E. coli clones that evolved that way. They're the same. However, I have a student, Alejandra, who just published this paper, who was interested in this question. Is there a difference between the E. coli lines that evolved by Rho or by RNA polymerase? And what she was really interested in is the idea of a thermal niche. The idea of a thermal niche is very simple. Is that any organism, you could put any organism on this chart, including humans, you have a temperature range in which you can evolve, or you can survive. And you might have a range in which, you might have an optimal temperature. My optimal temperature is about 80 degrees Fahrenheit. But for E. coli, that optimal temperature is about 37 degrees centigrade. Okay? We know that actually E. coli dies at about 43 degrees centigrade. So that's the upper limit. And its bottom limit is around 17 degrees. She was very interested, since we use temperature as selection pressure, she wanted to know, did we move this niche? Did it move up? And did it move the same for the two different pathways, RNA polymerase and Rho? Okay. Oh, that's a key point, sorry. And one way that you can ask that question is to ask, do they do more poorly? These evolved clones, do they have a bad fitness at the bottom end of the niche? I'll show you what I mean in a minute. Okay, so what we did is we took again our 114 clones, our bacteria, and we did fitness trade-offs at different temperatures. So at 37 degrees, it turns out that 26% of the clones performed just as well as the ancestor. Oh no, 26% of the clones performed worse than the ancestor. Okay? So when they evolved to 42 degrees, it meant that they weren't as good growing at 37 anymore. But that's only 26% of them. But if you go to a lower temperature, more than half of the clones don't grow as well as the ancestor. So in ability to grow at 42 degrees now, they've lost ability to grow at lower temperatures. So Alejandra really wanted to know, again, if there's a difference between Rho and RNA polymerase. And she did that by measuring the very extreme ends of the niche. And I don't want to go into too much detail, but she had to grow her bacteria over several days, and she saw if they actually persisted over several days, or was the temperature such that the population crashed. So she did these kind of measures at different temperatures, and I'll just give you an example of what the data look like. Here's all our evolved clones at 43 degrees, and you can see if you grow the ancestor at 43 degrees, it's very unhappy, and it doesn't grow very well. So she did this kind of assay at all kinds of different temperatures, but she could do it, for example, at 17 degrees, and she found all of our clones died, including the ancestor. If you do it at 18 or 19 degrees, you can see at 18 degrees, many of them survive, including the ancestor, but many of them die. And at 19 degrees, most of them live. So the question is, which ones are dying and which ones are living? And is there a difference between RNA polymerase and the Rho terminator, those two different adaptive pathways? So we did genetic associations. Our phenotype is whether it lives or dies, and we're associating with mutations. And we went through a linear regression, but here's the main point. If you have RNA polymerase mutations, you tend to die at 18 degrees. If you have Rho mutations, you tend to live. It's not always consistent, and that's okay because there's other mutations in these genomes. But it is very clear that these two different pathways differ in a very important feature. And that is, once they've evolved to 42 degrees, how well do they grow at low temperatures? They grow about the same at 42 degrees, but at low temperatures, there is a difference. The nice thing about E. coli is you can actually test these things by making single mutations and then doing the same work. I won't go into detail, but in this paper we had 1, 2, 3, 4, 5 single mutations. Our prediction was that these should die at low temperature, This should survive, and that's exactly what we found. The gray means it died. Okay. So, here's what we found on this experiment. Do I have a conclusion slide? Ha, no. Let me summarize this E. coli experiment. I think it's really cool. I think it's really fun. The first is, there were a limited number of genes that evolved in our simple experiment to high temperature. But the number of sites within genes that evolved is, at this point, unestimable for us. We know how many sites we see. We don't know how many possible sites there are that can lead to evolution. So in a weird way, evolution is constrained, 60 genes, and not constrained, a number of estimable sites we have no handle on. We do know, too, that there were two different pathways to evolution. So it's not completely replicable, even in this simple experiment. And we now know that these two pathways are not equivalent. They have different trade-off effects at low temperatures. Okay. Oh, maybe I do have a conclusion. Oh, evolution, I just talked about that, is remarkably flexible. Again, at the single base pair level in the simple experiment in the lab. But also, I think we all know it's remarkably flexible just by looking at things like Brassica oleracea. It's not clear to me how often genes are reused in crop domestication. It may be that every time it evolves, there's novel genes. But repeatability does depend on the scale. Are you asking about individual mutations, or are you talking about genes? So that's my first conclusion from these two very large evolution experiments, crop domestication and evolution. The other thing I didn't talk very much about is that interactions among genes are very important. I showed you about interactions between RPOB and rod genes, for example. For some reason, they tend to mutate in tandem. That's called epistasis. And you expect epistasis in E. coli because there's no sex and no recombination. But it's now becoming clear from studies like these that epistasis plays a very big role, interaction among genes in evolution. But we really don't know how big a role yet. The other thing that I think we probably all know, but sometimes we forget, you hear people talk about evolution and you think it's a very slow, gradual process. In fact, that's how Darwin envisioned it. But the point of fact is evolution can be very rapid. So dogs have been domesticated, I don't know, tens of thousands of years, but there's huge differences. Maize has been domesticated 8,000 years. Tons of differences. You can even see this very rapid evolution in the lab. So this is a picture of bacteria at very high temperature that is growing, and after one day it's very sick, another day it's very sick. Suddenly mutations occur, bam, it's happy again. Evolution can occur within days. It's a very powerful and fast process. And that's why it's fun to study. So let me finish by thanking you again, thanking Cordoba, and thanking the people who worked on all this stuff. Conchi and Esteban, I want to make sure. My wife, Becky, she's pretty nice too. I talked about all them, and these are some old friends and old collaborators who we've worked with over and over with in the years. So thank you very much. I'm happy to take questions if there are any.
Any questions? En español, en inglés, podemos traducir. One question.
Thank you for really interesting conference. So, in evolution, there is no experimental design.
Right. And?
I mean, according to your data.
Oh. Yes, so this question of replicability is not answered, really.
So it's pure chemistry plus environment.
I think there's just a lot of, okay, if you want to get to one place, high temperature for E. coli, or domestication, that's the selection pressure to increase the size of grains. My take-home message is there's a lot of ways to do it. In English, we'd say there's a lot of ways to skin a cat. That's an idiom. It means there's just many different ways through which evolution can act. And that's been an open question. And it's still, some would look at my data and say, no, it's very constrained. It's a hard question to get our heads around. But I would say it's remarkably flexible. That's my view of things.
Yeah, but in this temperature system with E. coli, I think it's a very simple.
Oh,
What you can do in the experiment.
That-
What do you expect from a combination of
Yeah.
stresses, environmental conditions? There must be mostly
That's-
dead and streets.
yeah. Well, let's look at that question exactly. Let's say we had E. coli in fluctuating temperatures, 42 degrees down to 17. We have two pathways, one that grows well at 42, not at 17. The other pathway does well at 42 and 17. We predict that only one of those pathways would survive. So all this fluctuation might actually limit evolution on us to some extent. Do you see my point?
Yeah.
Yeah, if you get a very heterogeneous environment. But again, that's the whole point. These are very simple experiments, and yet we're getting astoundingly shocking numbers of novel changes. It's a good question.
¿Más preguntas?
You say that the evolution is rapid.
Yes.
It may depend on the genome size,
Oh, yes.
if the organism is adapted to the environment,
Yes.
it is very well set up there, so it doesn't need to change.
Yes.
Or
You're-
what?
What I mean is it has the potential to be very rapid. And I think sometimes in evolutionary biology we forget that. You know, I come from the United States where people don't always believe in evolution. I'm sure you know that. And their argument is often that evolution occurs so slowly. How do we get so many species? And there's two problems. One is there has been a lot of time on Earth, and many people in the United States don't agree with that. But also that evolution can be very rapid. It's not always a very slow process. Very fast changes can occur. And we can see these, for example, the dogs. There's a gene that causes dwarfism. And so very rapidly, one mutation, boom, you get a very different looking dog. And sometimes that's forgotten. So that message was really not for you. That was for the public in the United States. Does that answer the question okay? Yeah.
Any other question? ¿Más preguntas?
Yes, in relation with the size of the genome,
Yes.
do you have any explanation for this?
No, absolutely none. Again, there have been some comparisons between species that suggest transposable elements can go crazy and cause big changes in genome size. So we thought that would be the cause, but it's not. It's these heterochromatic repeats. We don't know how they're replicated, why they're replicated, and why genome size should correlate with environmental characters, because that suggests genome size is important for adaptation to different environments. We just don't have an idea about that. And in fact, I think that paper was important just to even make the point that it does vary so much, there hadn't been many studies like that, and that it is predictable by environmental factors. But we have no idea why. That's a great question. You can think of, you could fantasize, but I have no real answer.
I got not a question but
Sure.
a comment.
Sure.
I would expect this room totally full of students, but probably it's not the case. So can we give them another opportunity so it is possible to have your presentation?
They are recording the
There.
presentation.
Yeah.
Then the presentation will be available,
But
I
they don't-
guess, in the webpage of the university.
Yeah, I mean, if you don't mind, if you can
I don't mind at all.
give it to
No.
Conferencia with us, I don't like movies very much.
In the webpage of the TIA3, in the...
Sí, ponerlo con
It will be
en
available.
PDF, que le quito lo que él quiere porque me parece que es una conferencia.
Yeah, absolutely no problem.
I have a comment because, aunque, bueno, I would say the
Sure,
Spanish
please,
person,
please.
aunque yo con él siempre he estado trabajando en la línea de plantas, de genómica de plantas, la mitad del laboratorio trabajaba en la evolución de bacterias. Y entonces, viniendo de una escuela de agrónomos y de cosas muy aplicadas a la agricultura, siempre desde el principio me pareció que la agricultura es experimental evolution. Porque, por ejemplo, nosotros tenemos una plaga y entonces el agricultor ejerce una presión de selección muy grande que es aplicar un fungicida. Y ese fungicida está en contacto con la planta durante años, años, años y años. Y tenemos esa presión de selección. Hasta que al final ese hongo, esa bacteria, adquiere resistencia. Entonces, cada sistema agrícola, o en cada caso, cada pareja enfermedad-planta, es un sistema de experimental evolution. Mucho más complejo porque además del fungicida tiene las condiciones ambientales de cada sitio. Pero me pareció enorme la aplicación de estos experimentos de evolución experimental en laboratorio para predecir lo que puede ocurrir, por ejemplo, cómo un patógeno puede adquirir resistencia en el campo. Y eso es algo que vi que en agronomía no se hacía, no se estilan este tipo de experimentos. Cuando creo que es el sistema donde tiene más aplicación, donde podría tener una aplicación directa. I am telling that even though I have been working all with plants, with you, the half of the lab was working with bacteria. And since the beginning, I thought that that agriculture is a system of experimental evolution.
Yes. Yes.
Because you have a crop and you have a pest or a disease, and then you are applying a fungicide during years. You have your own experiment for experimental evolution.
Yeah.
And that pathogen is mutating and trying to acquire resistance to adapt to those conditions.
Right.
And I was surprised that in agriculture anyone is conducting that kind of experiments, even though
Not that I'm aware of.
with those experiments you could predict the behavior of that.
There have been some, like for mosquito.
Mosquito?
There's a famous case where, you know, it's spread for pesticides and then it came back and they could actually find the gene that conferred resistance. That's the only one I know of. There may be more, but it's not been studied much. But it's a very open and interesting question, right?
Yeah, I mean every pear crop disease is a different experiment, and those pathogens are evolving.
Yes.
In contact with the host.
Yeah.
And maybe eventually some of them will become resistant, but we don't know that if they will become resistant because they fix the same mutation.
Yeah.
Or because they fix different mutations that are producing the same effect, that at the end is to survive the fungicide.
Yeah.
And I guess it's
an interesting question. It's a tough one though because it also involves interactions.
Yeah, we don't have so controlled conditions
exactly.
like in a lab. But I guess it's something that is happening now.
Right. Maybe fast. Maybe
rapid. Un poco. So there is here at the University of Córdoba, you know, Rafael de Prado's group working on herbicide resistance for long. So he has a very good collection of herbicide resistant weeds. I was collaborating with him for a while, and I remember perfectly a big controversy between two different opposite theories. So herbicide resistance is selection or is mutation plus selection. And somebody thought that probably selection, the biotype is there and is selected on the basis of the use of the herbicide. Because say, okay, it doesn't make sense to appear mutation selection in just 5 or 6 years.
Yes. This, in fact, so again, E. coli is a very simple system. We set it up so we would find new mutations. But this exact debate you're talking about is going on in eukaryotes. So there's been experimental evolution in Drosophila. And they're finding, they don't find many new mutations, it's just selection. Right? So there's that debate, and the question is how often is it new mutations, how often is it just selection? And what's the limit of selection without new mutations? It's still going on. In fact, it's becoming a bigger debate now.
It's the same, it's a matter of time.
Yeah, right.
One question, when at the beginning in this experiment in which you are evolving bacteria, you see that at the beginning, the first generations that are trying to grow high temperature, there is like a burst of mutations.
Yes.
At the beginning, the first generation, suddenly a burst, like an explosion of mutations occurred. And then after that there is selection.
Yeah, very soon after, right.
Then it seems that like a hard stress is able to produce at the beginning like an explosion of mutations.
So, you're getting at a question I would like to ask, and that is, no, it's a great, if you have a very hard stress, does the actual rate of mutation increase?
Yeah.
We don't know the answer to that. And that's, I think, a really interesting question.
I could expect that.
You would
because
expect
all the enzymatic machinery doesn't work properly. So
Yeah.
the number of errors
Yeah,
will be increased.
that's interesting. I think an evolutionary biologist would say, nah, it's going to be the same. But I totally buy your argument. I hadn't thought of it in those terms. I need a biochemist. That's what I need. Because things aren't working right. Yeah, so you're going to get more mutations.
I'm happy to go to the States. In Spain, there is no way to be financed to do this basic.
Well, we're having problems in the United States too. But I will say this picture is taken, this is sort of my weekly hike on the weekend. So this is about a mile from my house. So it is a nice place to visit, I will tell you that. Any other questions?
Any other? Okay.
Thank you very much.
Thank you very much.